Alright, in today's notes, we're going to take a look at Chapter 12 as a basic introduction to what the somatic senses are. So we take a look at what a somatic sense is. What it refers to are 5 basic senses: a sense of smell, sense of taste, sense of touch, sense of sight, and a sense of hearing. But in order to actually have those senses, we need to have a receptor site. So let's go back to the nervous system in terms of our reflex arc. Remember in our reflex arc that we had talked about, we needed some sort of a landing site for something to happen. And as soon as our landing site received and landed on a receptor, the stimulus is then going to be carried through a sensory neuron, eventually carried to the brain and spinal cord in the interneurons, and eventually is then sent back out motor neurons. All right. What we're looking at in this sense is we just want to focus in on this area here. What are those sensors? What are those things that allow the outside world or environment to stimulate that of a sensation, bring it into our brain or our spinal cord? So if I just kind of quickly get rid of this stuff, move our way into some of the notes, we're going to talk about how these sensory receptors are going to be working. Now, what they are is a specialized type of cell or multicellular structure that's going to be job is to collect or hold on, receive signals of all of the outside information. Their job is to stimulate then the neuron, the receptor neuron. So if we kind of shrink this up, again, remember we have some sort of a site, initiation site. This whole job, somewhere in here, there's going to be these little hair cells or who knows what, maybe just very, very sensitive structures are receptors. And if I have anything from the outside world coming in and landing on those receptors, they send the signal to that nerve pathway. They remember they're going to reach what's called the threshold. That's one of the things we talked about in the nervous system, the threshold stimulus. What amount of a particular thing do we need, sort of stimuli do we need before this impulse is actually going to be reached? Let's actually move our way on. Now there are actually, looking at here, we've got 5 different types of receptors that are going to be on the body. The first one is going to be referring to a chemoreceptor. Chemoreceptors are referring to then of the chemicals. Okay, chemo is chemicals. Pain receptors responds to tissue damage. So anything referring to tissue damage, tears, swelling, that's where we get our sense of pain. Thermoreceptors, thermo, like a thermometer, is referring to temperature. Anything with that is your temperature. Mechanoreceptors, mechano refers to movement. Let's get a different color in here. Mechano is referring to movement. Okay. And then the photoreceptors are referring to photons. When a photon, if you've had physics, things that you remember are what those are light energy. So it's going to respond to light. Now, in certain areas where we might find some of these, after the 5 different senses, so if we kind of go back and think about our 5 different senses that we have, we've got 1, the sense of touch, smell, taste, hearing, sight. So those being our 5 different senses, they're going to have to be referring to some of these types of receptors that we're going to have in the body. Now, some of the quick and obvious ones that we could talk about is like these two, the sense of smell and taste. How do we get the different senses of smell or different tastes are based upon different chemicals that are going to be stimulating those. The perfumes that are in the air, the saltiness, if it's got sodium chloride, that the chemical in there, if you taste salty, if you taste the glucose, meaning you taste something with carbohydrates, it's sweet. All sorts of different degrees are based upon chemicals, and those are the chemoreceptors that we're referring to here. And obviously, when we look at this guy, a photoreceptor referring to light is going to be something that we're going to be looking at in the case of sense of sight, things we can look at. In terms of hearing, that's going to refer to some of the mechanical forces in terms of sound waves. We'll talk about those. And then looking at touch, you're going to have a multitude of them. Thermoreceptors is going to be a big one. Mechanoreceptors will be a big one there. And we'll see those here shortly. So, in looking at this, let's shrink this a little bit so we can see it all. Now, a sensory impulse, what this is going back to our our reflex arc. Okay, you have some sort of stimulation on a receptor site. Look at our receptor site. What may it be? Something in the skin. Maybe it's in the eye. Maybe it's in your nose. Maybe it's on your tongue. Based upon whatever type of sensation pathway we're working with, needs to have an initiation site. Now, once that stimulation, whatever that foreign thing is, lands on that site, you are going to reach that threshold stimulus, the minimum amount of something needed. Now, if we really go back to the nervous system, we talked about if I have to increase that amount of sensation or increase that amount of stimulus, what I'm doing is summation. Now, if I have that actual stimulus being created, again, we have our structure, whatever it might be, lands on that stimulus. It then has the signal to start our nerve impulse. Now, in order to bring it into my body, I am going to go through that sensory pathway, sensory division of the peripheral nervous system. So it's a great electrical current that is generated. Now, in this unit, I'm not going to go back to the polarization, depolarization, repolarization of the impulse. That's something we covered in first quarter. What I do want you to remember, though, is the pathway that these guys are going to be taking. So if we have our receptor site here, remember we go from receptor to sensory neuron to interneuron to motor neuron to effector. Okay? That's the generalized pathway that these sensations are going to be taking place. Okay, so if I look at our steps here, what we see, again, we've got that stimulation, the great electrical current is going to be stimulated, a receptor is part of the neuron, we've talked about that. The membrane potential is going to reach action, so it's going to be stimulated, polarization, depolarization, repolarization. And if the receptor is not part of the neuron, you're going to have something in some sort of a connection site that is going to eventually stimulate that neuron. What it all comes down to is you are going to get some sort of a feeling. Now, is that feeling a sense of smell? Is that feeling a sense of taste? Is that feeling light coming into your eyes? What is it? What that feeling is a sensation. It is the time in which your brain actually interprets the impulse. Okay, so sensation is the feeling that your brain gets when your brain is actually interpreting an impulse. Now, sometimes we can get an impulse that lasts a long time. And one that lasts a long time, you kind of get used to it and kind of goes away. Now, take, for example, your nose. Can you see your nose? If you cross-eye yourself, you can take a look and you can see your nose. But we get used to it. We look past it. Another example is when you're sitting at a ball game. Maybe there's a net in front of you. I'm thinking baseball now. You got the fence in front of you. You can't see a whole lot, but as soon as you sit there for a long period of time, you start looking through the fence. You get used to that. One other one, maybe in the sense of smell. Okay, long bus rides, when you're on your field trips, you know that somebody decides he needs to pass gas. As they pass gas, you're in that car for a long period of time or the bus for a long period of time, you get used to that smell. But if you were to go back to that bus or the car ride, you know that that smell is still kind of lingering inside there. In terms of class, I'll give you one last example, dissection. When you're doing dissection, you get used to the smell. But if you were not, and you walk into the classroom, the smell can be overpowering. Okay? So, in this term, what we do when we get used to something, we adapt. Adaptation based upon the senses. Okay? The smell receptors undergo a lot of sensory adaptation. So now we look at our types of somatic senses that we have. Senses are going to be ones that are going to be associated with the skin, joints, muscles, and viscera. Viscera, again, refers to organs. Now, there are three different types of groups that we're going to see. Some on the outsides of the body, so we're referring to exteroreceptive senses. This is where you see in the body's surface. Touch and pressure receptors, pain receptors, thermal receptors are going to be found there. You're going to be finding some within the joints. These are proprioceptives, and these are going to be seen when tendons, connections of muscles to bones. You also see them in ligaments. And the last one is an organ, visceroreceptive senses. Now, if you are having organ pain, you may not know it. These visceroreceptive sensations are things that are going to travel those nerve pathways and tell you that your body is aching, it's sore. So if you ever had like a kidney stone, well, how do you know you actually have a kidney stone? Why is it making you feel sore? Why is it painful? If you're a gallbladder, you're a gallstone, what is actually making that sense happen? Okay, so we have these receptor sites in those organs that actually stimulate some sort of changes within inside the organ itself. Now, if I look at the first types of those type of somatic senses, we're only going to do this first section today, and that's dealing with touch and pressure senses. We've got a few of them that we're going to cover. The first one being free nerve endings. Free nerve endings are actually going to be closest to the surface, and they're going to be common in epithelial. They're going to work their way all the way up towards the surface of their skin. Now, they're not going to be in the outermost layer, but they're going to be close, and they're going to be able to sense very, very light touch and light pressure. The second one down is the Meissner's corpuscle. This is abundant in hairless regions. Think of your bottom of your feet. Okay, look at your underside of your wrist, your palm area. Light pressure, you can sense that. Any sorts of pressure you have, Meissner's corpuscles are going to be around those areas where there's a lot of hairless regions. Now, heavy pressure, really pressing down on something, is going to be found and detected by the use of this Papsinian corpuscle, which is going to undergo that heavy pressure. Understand that, and that's going to be in deeper tissue. So if I take a look at this illustration here, illustration will help us identify it. Bring it up a little bit. We have, here are our free nerve endings. There is the base of your epithelial tissues here. Now, these free nerve endings, let's erase this, are going to wiggle their way on through this epithelial, and they're actually going to be inside of the skin layer. Now, just below that epithelial, in the dermal layer, you're going to have these larger masses of receptor cells. The larger masses of receptor cells are going to be these guys. Those are the Meissner's corpuscles, typically in hairless regions up near the surface because they detect light pressure. Further down, you're going to have bigger ones. Those ones are going to be undergoing heavy pressure. Understand those are the Papsinian corpuscles. Okay. In terms of temperature, thermal receptors, these are going to be receptors that are going to be sensitive to different types of temperature. Warm, cold, and when it becomes too warm, you start to have tissue damage, pain receptors, and when it's too cold, you have tissue damage, pain receptors. For example, going outside right now, it's awfully cold outside. So if it is below, look at the cold receptors, 20 degrees Celsius or 68 degrees Fahrenheit, you're going to start to get the sensation of cold to the point now where you saw the last couple of days, it's 5 below outside. It is way too cold. You're going to cause a whole bunch of tissue damage, and you get to the point where you become unresponsive to that cold, and you could freeze to death, leading into hypothermia. Right? Warm receptors, what they're sensitive to, you will want to remember this in terms of degrees Celsius and in Fahrenheit. You're going to see you're sensitive to above 25, which is 77 degrees Fahrenheit. So anything above 77, you're starting to feel warm. Anything below 50, you get the sensation like, whoa, we're getting kind of chilled, too cold. Okay, so our temperatures between 50 to 68 is our chilled sensation. Between above 77 is the warmth. So between 68 and 77 is kind of our nice normal temperature we'd like to be at. Now again, we're in Minnesota. We get used. We undergo sensory adaptation. We get used to the chilled temperatures versus somebody like Florida who gets used to the warmer temperatures. Okay. All right, so that is where we're actually going to be stopping for today. We've talked about different types of receptors that we're going to have within our body, as well as where they're going to be located in the body. So you can go ahead and start working on the very first lab. The lab that you're going to be working on is Lab 31 on receptors and the somatic senses.